Good evening, everyone. Firstly, I'll introduce myself. So, I'm Dipanshi Avasthi, your TA for the session of Structural Biology course, and we'll be having problem solving session for the 12 weeks. So, in this session, we'll be discussing your doubts and any issue which you face in the week content. So, that thing we'll discuss. Along with this, we'll also go through the entire week content in which we'll see that what you have studied in the entire week. So it will be like a short summary of the week one topics or respective week topics, and then we'll also see the new literature or what are the new things which are going on. And along with that, we'll practice some MCQs with that. And other than that, if you have any doubts, then you are welcome to ask them. So in case of any query or any doubt, you can raise your hand or type in the chat box, or you can unmute. And when in the end I'll discuss the questions with you, so in that I expect that you also respond so that it will be one to one or it will be interactive session. Okay, so that's it about. So now I'll start. So this is the session layout. So in this, firstly will be the introduction about various topics which was covered in week one, and then we'll practice MCQs, talk about the current literature, and I'll take your doubts. So before proceeding, I'll just confirm that are you guys able to follow? Okay, so shall I start? Just confirm in the chat box because I can see that four people have connected. So any one of you can type in the chat box that should I start? Is there any issue? Are you able to see my screen, right? Okay, so I'll start. Um, I think you people are a bit shy and not replying time. Okay, so we'll talk about the introduction to structural biology. That what is structural biology? This course is about the structural biology. So it is a study of molecular structures and the dynamics of macromolecules, that how they are present in the body and how they interact with each other, how they are present dynamically. Means they are not constant or fixed. They are constantly interacting, moving. So all those things come under structural biology, and particularly we focus on biological macromolecules such as proteins and nucleic acids. Although there are other macromolecules also available, but our main focus will be on the proteins and nucleic acids, and we'll also see that how their alterations in the structures affect the functionality. Along with this, structural biology also includes the principles of molecular biology, biochemistry, and biophysics. So there are multiple branches which are interlinked. So it is an interdisciplinary study in which multiple branches come together and they understand a macromolecule and understand a thing. So this is about the structural biology. Now the question comes that why do we study structural biology? So by studying this, we can understand the protein architecture, that what is the structure of a protein, how it is arranged. Second thing is macromolecular interactions, that how this protein is interacting with other macromolecules. Because in our body, there are multiple proteins and many other things, nucleic acids, proteins, carbs, lipids. So these all things interact with one another and perform some functions. So all these things can also be understood with the help of structural biology. So if we know the structure of a particular macromolecule, then we will know that with which particular molecule it will interact, at what position it will interact, and what could be the result. So with this, we can understand the interactions. Now we can also understand the membrane organizations. So what's the, I'm sorry, I think there was some network issue. I got disconnected. Okay, so I hope now my slides are visible. Okay, so I'll just again explain from why do we study structural biology. So just confirm for once that are you able to see my slides? Okay, thank you so much and so sorry. So thank you. So yeah, so I was talking about that why should we study structural biology? So the first thing was the protein architecture, that how the what is the structure of proteins, how they are arranged. So this we can infer by studying the structural biology. Second thing is macromolecular interactions. So these are multiple macromolecules, proteins, carbohydrates, lipids. So all these interact with one another and they perform certain reactions. So by knowing the structure of particular macromolecule, we will be able to know that at which site it is interacting with other macromolecule, and with that we can predict its functions. So those inferences can be drawn if we know the structure. So this is also one of the advantage of studying structural biology. Third thing is membrane organization. So as we all know that when the structure of membrane was studied, so it was known that it is made up of phospholipid bilayer. Along with this, there are multiple proteins which are embedded in this uh, bilayer membrane. So some proteins are integral proteins which are spanning throughout the membrane. Some are the external extrinsic proteins which are present on the surface of the membranes. So these proteins have some functionality. They maintains the structure of the membrane and they decides that what should go inside the membrane and what should flow outside the membrane. So there are multiple proteins which are arranged on the membrane and they are helping in organization of that membrane or organization of the cells. So different cells have different proteins present in their membranes. So in that way, they are helping in the membrane organization. So we can study membrane organization by understanding the structure of the proteins, because you can see here that there are certain proteins which are polytopic transmembrane proteins. That means they are passing multiple times throughout the membrane, whereas certain proteins are only biotopic membrane proteins. So in that way, we can understand that organization with the help of structural biology. Next thing is protein synthesis machinery. So how the proteins are synthesized. So how ribosomes are the bodies which are synthesized of small nuclear RNA along with the protein complex. So how they are helping in the synthesis of proteins. So that can also be studied by looking at their structure. That how different units of ribosomes are joining to one another, how the another, for example, tRNA acetyl synthetase enzyme is coming, it is bringing amino acids. So how different amino acids are linked to one another to synthesize a protein. So in the back end, proteins are helping to synthesize new proteins. So in that way, by looking at the structure of the proteins, we can understand that what is the synthesis machinery of the proteins. So in that way, structural biology is also helping in that. Along with this, it also helps in understanding the interaction with solvents. So how a particular macromolecule will interact in solvents. So solvent could be anything such as water or other things. So that can also be studied with the help of structure. So if we know the structure, we will be able to know that which uh, parts are exposed and which are more prone to interact with the solvent. So means which molecules are present on the surface. So in that way, we can understand the interactions. And then protein conformations. So conformations are different structural orientations, you can say, that a protein can adopt. So with the help of structural biology, we can understand that in which conformation a particular protein is present, whether it is a closed conformation or it is an open conformation. So there are certain enzymes which bind with substrate, and there is a possibility that they undergo their change in their conformations. So all those things can also be studied if we study the structure of a protein. So in that way, these are different fields which can be explored with the help of structural biology. So now we'll move to the history of biological inventions, that how we land on to structural biology. What was the history, that how it all began? So I'll just check. Okay. Yeah. So now, as we know that biology, it is the study of living organisms and their vital processes which are undergoing in their bodies. So there are different means branches, or when it began. So firstly it was a natural history, which is the observation based. So in the primitive time, when there were no microscope, nothing was there, so at that time everything was based on observations. So in that you were believing what you are seeing, so seeing is believing. So this quotation comes from natural history time, in which the study of plants and animals was only relied on observations. So the records show that Babylonians, Egyptians, Chinese, and Indians, they had made numerous observations because they were performing cultivation, agriculture, or some medical practices. So whatever they used to observe, they used to record it. So in that way, whatever is available on the basis of observations, that is stored in the form of natural history. But later on, the Greek contributors came, and means slowly and slowly they bring some classifications or some technological advancements happen, certain inventions were done. So with that, the more and more information was gained. So starting with Aristotle, he gave the systematic classification of animals based on certain principles. So when he observed animals, he saw that what are the principles that distinguish animals from the other organisms. So in that way, he has given the systematic classification of animals. And similarly, Theophrastus, who was the student of Aristotle, so he performed the same systematic classification but for the plants. So advanced botany was given by him. And then the concept of atoms was given by Democritus, who checked that what is atom, and he mentioned that all the matter which is present, it is composed of small indivisible particles, atoms. So at that time, not much information was known that what is present inside the atom. So according to them, atom was the smallest unit, which was the indivisible particle, and infinite universe, it consists of atoms and void spaces. Then Thales of Miletus, he came and he believed that universe is driven by a creative force, which he named as physis. And then certain other scientists came who mentioned that this universe is composed of certain elements. So Thales said that water is the basic element with which the matter is constructed. Then Anaximenes came and he said that air is the basic element. Then Heraclitus came and he said that fire is the basic element. And then Empedocles came and he said that not just water, air, or fire individually, but they all are combined along with earth element and they constitutes the matter. So in that way, these four elements came into picture, that how the matter has been synthesized. And then humor's theory came, in which the Hippocrates school believed that there are four humors, such as blood, pledge, yellow bile, black bile, and they also link them to different elements. That blood stands for the air element, yellow bile stands for the fire element, phlegm stands means it is linked with the element water, and black bile is linked with the earth element. So in that way, they are linking elements with the humors. But there are certain limitations with the ancient Greeks, such as there were no experimental tools to verify this hypothesis, and microscopic inventions were also not done, so animal and plant structures could not be understood at that time. So later on, certain more advancements happened, and then visualization instruments came into picture. So it was previously known for about 2000 years that the glass has the magnifying power. That means it can adjust. With glass, we can adjust the magnifying power. We can change the magnifying power of different glasses. So Romans invented the glass in the first century AD. And then Hans and Zacharias Janssen were the ones who discovered compound microscope. So this was the first microscope, which was the compound microscope discovered, and it has 9 times its magnification, and it was low utility, so it was not having that much utility. So later on, Anton van Leeuwenhoek came in 17th century, who made practical microscopes which had up to 270 times magnification, and he was the first one to see bacteria and protozoa. So with Jensen and Antony van Leeuwenhoek, when the microscope came, then this discovery process means then this identifications and studies, they advanced, because now we have a medium with which we can see different objects. So then advances in microscopy happened, in which when Robert Hooke came and he used these Anton van Leeuwenhoek microscopes, and he coined the term cells. So when he was observing the bark of a tree, then he observed that there are small units. So they are like cells. So according to him, the cells are like the small rooms which are present in a bark. So he called them as cells. And it was also published in his book Micrographia. And later on, there were some chromatic aberrations, so which was the problem with those microscopes. So they were also later on resolved with using the achromatic microscopes in 1830s. So in this way, different advancements happen. And after the discovery of cells, Sheldon Schwann came and they mentioned they have given the cell theory in which they said that plants are composed of cells according to Sheldon, and according to Schwann, they said animals are also made up of cells. So later on, they have given a theory in which there are multiple points, but this was their main observation. And then Rudolf Virchow was another scientist who have given this quotation that omnis cellula a cellulae, which means that all the cells they arise from pre-existing cells. So now we'll see that what cell theory means. So in cell theory, when Sheldon and Schwann has given this, so they said that all the living organisms are made up of cells. And second thing was that these cells are the basic unit of life. That means these are the smallest and the basic unit. And these cells arise from pre-existing cells. So this was said by Rudolf Virchow. And then hereditary information is passed from the cells. So they are also passing on hereditary information, which they are having. So at that time, the knowledge of DNA or genetic material was not known. So they said there is some hereditary information which is passing on by these cells. And then they said all the cells have basic chemical composition. That means they have similar composition to each other, and energy flow occurs within the cells. And there is some energy flow which is undergoing. So these are the points of cell theory given by Sheldon and Schwann. And later on, after the cells have been discovered, the scientific attention moved to the processes which are happening inside the cell. Now, when you have explored the cell, you have seen the cell. Now, the next thing was that what's undergoing inside the cell? What are the processes which are taking place? What are the molecules which are present inside the cell? And then in 19th century, two other developments also happened, which also influenced this shift. So one was Charles Darwin theory of natural selection, and George Mendel's laws of hereditary. So Mendel also given the laws of hereditary. So with this, the attention was again shifted. So according to theory of natural selection given by Charles Darwin, he has published this theory in the book Origin of Species. And the key ideas of this theory were that the variations in the nature exist and certain variations are advantageous. So means as we have also seen that many people vary from one another, different organisms vary from each other. And within the same, even if we talk about the same taxa, then also different species vary from each other. So these variations are helpful in the nature, and some of these variations are also advantageous. And then struggle for existence favor the organisms better adapted to their environment. So which is also known as survival of the fittest. So as the environmental condition changes, the one who is able to adapt to those changes with the variations, he will survive, and the other one who will not be able to means cope with those situations, they will die. So in that way, only those will survive which have better adaptations, and advantageous traits are passed to future generations. So the traits which have helped them to survive, they will be further passed on to future generations so that they can also survive. So this is about the theory of natural selection. And then about laws of hereditary by Johann Mendel. So in this, he has studied a pea plant in which he means based on this theory, he has performed fertilization experiment, and he has formulated three main principles. First one was law of segregation, which says that paired hereditary units they separate during the gamete formation. And then second law was law of independent assortment, so in which he said that the traits which are inherited independent of one another. And then law of dominance, in which he said that in hybrids, the trait which is dominant, it will always dominate over the recessive trait. So the one which is dominant, it will express and it will suppress the means recessive one. So these are the laws of hereditary. And this is just a brief info, and I'm not going into the detail of these laws. So before moving on to genes and genetics, just let me know that are you able to follow, or am I going very fast? Okay, so yeah, okay. So let's start with genes and genetics. So after Mendel's discovery, he has said that there are certain factors which he later on named as genes, so which pass on from progeny to progeny. So Mendel's work gained recognition in early 20th century, and key milestones were that in 1905 William Bateson he coined the term genetics, and in 1909 Wilhelm Johansen introduced the term gene. So although Mendel is referred to as father of genetics, but when he performed this discovery, then he has not coined the term gene or he has not coined the term genetics. These were later on means coined by other scientists, but his work was quite interesting and he was the one who performed means interesting discoveries in that pea plant. So with he has laid a foundation for the genetics. So that's why he is referred to as father of genetics. So then the next thing came to check for the gene location in the cells, that where are these genes present in which particular part of the cell. So it was identified that the genes are present in the nucleus, and the term nucleus was given by Robert Brown in from 1825 to 1838. So he has coined the term nucleus. And then Walter Fleming, he described chromosome movement during the cell division. So he was a cytogenetist. So he studied the means when he was studying the cell, so he means found that there is the movement of chromosomes during the cell division. So when cell divides, the chromosomes are moving from the mid part towards the polar poles, or means he has studied that chromosome movement. And in 1915, Thomas Morgan came and his team confirmed that genes are the physical objects present on specific chromosomes, and these are the fundamental unit of hereditary. So also linkage concept is also described by Thomas Morgan, because when Mendel was performing experiments, he has given a law which is known as law of independent assortment, in which he said that two traits, they are independent of one another. So means he said they independently get inherited, but Thomas Morgan found that these genes, their distance is very important. So if they are present close to each other, then there is some kind of linkage which is operating. So the assortment of trait is not completely independent. So that's why he said that these genes, they are the physical objects which are present on specific chromosomes, and he also established the chromosomal theory of inheritance. So previously, this chromosomal theory of inheritance, it was given by Walter Sutton and Theodore Boveri, but this theory was not experimentally proven. So with the means experiments performed by Thomas Morgan and Drosophila, so he established this chromosomal theory of inheritance. And now the focus has shifted to cell biology, that means what is undergoing inside the cell. So with the advancement of microscopy, we were able to visualize the cells as well as the internal structures, that what is present inside the cell. Means, for example, different organelles which are present. For example, in the previous slide, we have seen that how Robert Brown has also investigated, he has identified that there is some nucleus which is present inside the cell where the genes are present. So in that way, with the help of light microscopy, so the compound microscope which was discovered by means Anton van Leeuwenhoek. So they comes under light microscopy, in which they need light for visualization of certain things under the slide. So it marked the beginning of structural cell biology, in which the cell structures were studied. So it allowed the scientists to study the cell structures and identify basic cellular components. However, its resolving power was limited. So resolving power means that means how far you can see two elements which are present close to each other. So if the resolving power is strong, that means even if two objects are present close to one another, still you will be able to see them like means far away from each other. So you will be able to clearly see them. But the limitation of light microscope is that its resolving power was lesser. So by the wavelength of visible light, so which due to which you could not visualize the finer details of the macromolecules. So later on, when advancement happened, other microscopes also came. So the biggest advancement was by the electron microscopes. So moving from light to electron microscope, there were other microscopes also which were present, but means they comes under the category of light microscopes only, where they try to improve the resolving power by either phase contrast or fluorescent microscopy. There are other types of microscopy which comes under light microscopy. So later on, the electron microscope was discovered, in which Instead of using light, they use the electron waves. So with much shorter wavelengths and electron microscopy, it provides highest resolution as compared to the light microscope. So with this, we can visualize the cellular and subcellular structures in detail. And it also includes the macromolecules such as proteins and nucleic acids. So with this, we were also able to see the macromolecules that are present inside the cell with the electron microscopy. So here is the distribution. So with the eye, you can see the objects which are up to 100 micrometer in size. So up to 100 micrometer can be seen with the naked eye. Then with the help of light microscope, you can see from up to 1 millimeter till 100 nanometers. So all those things are visible with the light microscope, such as your hair follicle, blood cells, bacteria. These can be seen with the light microscope. But if you want to see the viruses, DNA, glucose, atoms, so these cannot be seen with light microscope as they are smaller than 10 nanometers. So to see them, electron microscope can be used. So electron microscope provides you the ability to see the objects which are from 10 micrometer till zero point 0.1 nanometer small. So with the help of electron microscope, we were able to see the macromolecules which fall in range from 10 nanometers to smaller. So here is also the distribution that what can be seen with the electron microscope. So electron microscope is also two types. One is scanning and another is transmission. So they vary in the principles and theory. So with the help of scanning electron microscope, you can see till 10 raised to the minus 7 meters, which means includes the organelles. But if you want to see later on to the level of macromolecules such as DNA or small atoms, then we need transmission electron microscopy. So which is the better version of electron microscopy. Okay, so now we'll look into the basic macromolecules of life. So till now we have covered the concept of genes, genetics, and cell structure and what are the different components inside the cell. Now our focus has been shifted to macromolecules because now TEM or we can say transmission electron microscope came, which provided you the ability to see the macromolecules. So now we'll look into the what are the macromolecules which are present inside the cell and what are their functionalities. So now we'll discuss about them. So what are the basic macromolecules? So these are essential for the survival and growth of an organism, such as plants and animals. They obtain them from external sources. From food, they get these macromolecules. And then this term was coined by Hermann Staudinger in 1920, who proposed that means they form by the covalent linking of smaller molecules. So he said that these macromolecules, they are formed by the covalent linking of the smaller molecules and they play vital roles in the cell structure and its functionality. So that's why it is important to study these macromolecules because they are governing the function as well as structure of the cell. So if we want to see the classes of biological macromolecules, then there are four major classes, among which the first class is of the carbohydrates. So as the name suggests, carbohydrates. So these are the I mean, macromolecules which are composed of carbon, hydrogen, and oxygen, and they serve as a fuel and building materials. So building block of the cell, and it includes monosaccharides as well as polysaccharides. So in the later slides, we'll read a little bit detail about carbohydrates, but for now, monosaccharide means that it is having a single unit of the means single sugar unit. And if there are multiple sugar units connected to each other, it comes under polysaccharides. Now the next class is fats or lipids. So these are also constructed from glycerol, which is a 3-carbon alcohol, and fatty acids. So these are two of the building blocks of fats. And there are two types of fats, such as saturated, in which there are no double bonds, and then unsaturated, in which there is a double bond, and this double bond causes the bending in the chain of the fats which is present. So due to which you have seen that these saturated fats, they are means at the room temperature, they are solids, whereas unsaturated fats, they are liquid at the room temperature. And then nucleic acids. So nucleic acid is a third class of macromolecules, and these are the polymers of nucleotides. So nucleotides are further composed of nucleoside plus phosphate group, and nucleosides are composed of pentose sugar and nitrogenous base. So in the later slides, we'll understand this. And then this nucleic acids, there are two types of nucleic acids. One is DNA, another one is RNA. So DNA is deoxyribonucleic acid. So this naming is given on the basis of sugar. So if deoxyribose sugar is present, then it makes DNA. If ribose sugar is present, it constitutes RNA. And rest are the nitrogenous bases. So in DNA, there will be adenine, guanine, cytosine, and thymine. Whereas in RNA, you will find adenine, guanine, cytosine, and in place of thymine, there will be a uracil as a nitrogenous base. So this is the difference between the DNA and RNA. And then the fourth class is proteins. So proteins have diverse structures and functions. They provide support to the cell, storage, transport, as well as defense. So antibodies are also the proteins which are helping in defense. Then for transportation also the proteins are responsible, as we have seen previously that in the cell membrane there are multiple proteins which will also help you to transport the things in and out of the cell. Along with this, there are certain proteins which will also bind to other smaller molecules and help you to transport them to the other organelles or other places in the cell. So they account for over 50% of the cell's dry mass. So they are the major constituents of the cell, you can say, and they are built from amino acids. So amino acids are the smallest units which combine together to make proteins. And their basic structure is that they contain an amino group, one carboxyl group, and a R group, which is an alkyl group. So depending on the type of amino acid, it might vary. Okay, so now we'll talk about the carbohydrates. But before moving to that, do you have any doubts till now? Is there any doubt? Okay, thank you. So now let's talk about the carbohydrates. So these carbohydrates are composed of carbon, hydrogen, and oxygen, and they have general formula as CH2O n times. So depending on the number of carbons in the sugar, the formula will vary in this way. So means if there is only one carbon, then there will be two hydrogen and one oxygen. If there will be two carbons, then you have to multiply each term with n number. So in that way, you will be able to find out the formula of that particular carbohydrate molecule. So these are the primary source of energy for living organisms, and they also serve as the structural components in the cells. And it includes sugars, which are monosaccharides, starches, which are polysaccharides. So starch is a storage carbohydrate used for the storing food. Then fibers, and they play essential roles in metabolism, energy storage, and cellular structure. So carbohydrates can be divided into monosaccharides, oligosaccharides, and polysaccharides depending on the number of sugar units which are present. So monosaccharide, mono means one. So when there is only single sugar unit, it is referred to as monosaccharide. Oligo means multiple. So from 2 to 5 units are present, then they comes under oligosaccharides. And if more than 5 or greater than 10 units are present, then they comes under polysaccharide. And depending on the functional group, so it is divided into aldoses and ketoses. So which functional group is present in the sugar? So these we will see their structure in the next slide. Don't worry about it. And then number of carbon atoms. Then the naming can be trioses, in which there are three carbon atoms. Tetroses means there are four carbon atoms present in the sugar. Pentoses means there are five carbon atoms which are present. Hexoses means there are six carbon atoms, and heptoses means there are seven carbon atoms present in the single sugar unit. So means, for example, in glucose we have six carbons, so it comes under hexoses. So this is just a nomenclature that how you name a particular monosaccharide depending on number of carbons it contains. Then in oligosaccharides, there could be disaccharides in which only two sugar units are linked to each other, such as maltose, lactose, sucrose. Or there could be a trisaccharide in which three sugar units are linked, such as raffinose. Or there could be tetrasaccharide in which four sugar units are linked. And in case of polysaccharides, we have homopolysaccharide or heteropolysaccharide. So homopolysaccharide means in which similar type of sugar units are linked to each other again and again. So there are multiple sugar units, but they all are same. So they are homopolysaccharides such as starch, dextrin, glycogen, cellulose, inulin. Whereas heteropolysaccharide means if other than sugar, some other molecules are also present in the multiple units. If there are some other molecules, other bonds are also present. So it comes under heteropolysaccharide such as hyaluronic acid, heparin, chondroitin sulfate, dermatan sulfate, or keratin sulfate. So these are heteropolysaccharides. So these are the structures of monosaccharides versus disaccharides. So as you can see, these are the single sugar units. And if there are two sugar units, then they comes under disaccharide, which will fall under the oligosaccharide category. So for example, in case of sucrose, the glucose and fructose are linked to each other to constitute sucrose. Whereas in lactose, galactose and glucose, they combine to each other and form lactose. So now, as in the previous slide, we have seen that there are, means depending on the functional group, the monosaccharides can be called as aldose or ketose. So aldose means if there is a aldehyde group, which is CHO. So this is a means sugar which is composed of three carbon units. So in its terminal, if there is aldehyde group which is present, so it is referred to as an aldose. But on the other hand, if it has a ketone group, that means instead having the terminal double bond O, if it has that terminal double bond O in the middle, which acts as a ketone group, then it is known as a ketose. That means a sugar which contains a ketone group, and aldose means a sugar which contains a aldehyde group. So this is for the sake of representation, it is a sugar which is triose, that which contains three carbon units. So in means we can also expand it for the other sugar types. So for example, the triose is the glyceraldehyde. So it is also known as glyceraldehyde, in which this is a type of a aldose sugar in which aldehyde group is present. And if we talk about the pentose sugar, then this is the ribose, which contains five carbons. And if it has the terminal aldehyde group, then it also comes under aldose. Similarly, glucose is also aldose sugar in which there is a terminal aldehyde group is present. So it is a hexose, that means it contains six carbons. Now there is another thing which is quite confusing, which is the D and L configuration of carbohydrates. So after functional group categorization, next thing comes the configuration, that either it is D configuration or L configuration. So how to identify this? So in this case, you have to locate the chiral center which is farthest from this C double bond O. So in this case, I am only considering the aldose sugar. So similarly, ketose will also behave the same way. So you have to find the chiral center. So chiral means you have to find a carbon which is attached to four different groups. So in this case, we cannot select the last carbon because it is attached to two hydrogens. And these two hydrogens are same. So in that way, it is not a chiral carbon. So the next chiral carbon is the second carbon, and it is farthest from this terminal aldehyde group. So we will select this. So on the basis of this chiral center, we will look for the OH group. So if the OH group is present on the right side of this chiral center, then it is referred to as a D sugar. Whereas if the OH group is present towards the left side of the chiral carbon, then it is referred to as a L sugar. So these are also referred to as the enantiomers. That means they are like the mirror images of each other. So in this way, D sugar and L sugar. Okay, so let's understand what enantiomer means. So enantiomer is one of the two molecules that are mirror images of each other, but they cannot be superimposed onto each other. Much like how your left hand and the right hand are the mirror images of each other, but they cannot perfectly align on top of each other. So these molecules have same chemical composition, but they differ in their spatial arrangement. So they both have exact same means constituents, same number of carbons, same number of hydrogens, same number of oxygens, but they only vary in the arrangement of the OH group. So in one case, OH group is present on the right, in the another, OH group is present on the left side. And these structures which you are seeing, they are also referred to as a Fischer projections. So there are multiple ways to draw the carbohydrates. For example, if you draw them in the form of linear chains, then they are known as Fischer projections. And other than this, you can also draw these, means glucose can also be drawn in a cyclic form as well. So for example, in this case, you can also draw them in a cyclic form. So in this way, these are the aldoses and these are the ketoses. So in mostly in the case of ketosis, you will observe the pentose ring. And in case of aldoses, there will be a hexose ring. So this is the cyclic representation of these uh, monosaccharides, you can say. So in cyclic form also they exist. So in cyclic form, they have two variants. One is alpha form and one is beta form. So when you call alpha, so in this case, the carbon which is lying next to the oxygen, which is responsible for cyclic structure formation. So you'll see the carbon which is next to this oxygen, and with this carbon, this will be a particular center which is considered for observing the OH group. So if OH group is present below the means towards the below the this particular carbon, so below this means, you can say downward to the O group, then it is referred to as alpha glucose, whereas if it is present towards the same direction to which the oxygen is present in the cyclic form, so it is referred to as a beta glucose. So these will be helpful when we will see the polysaccharides such as starch, cellulose. So there we will, when we study the bonds, that alpha 1,4 linkage, beta 1,4 linkage. So in that way, this orientation is helpful. This will make you aware that why we are calling them as beta 1,4 linkage or alpha 1,4 linkage. So that is determined by the position of this OH group, that whether it is present towards the same direction of the oxygen present in the cyclic ring or opposite to the cyclic ring. So if opposite, then it is referred to as alpha form, and if it is towards the OH, towards the oxygen, then it is beta glucose, and these are referred to as the anomers. So in case of D and L form, we have learned the term enantiomer, that means mirror image, whereas this is an anomer, which means different forms of the sugar molecule which differ in the configuration of hydroxyl groups attached to the anomeric carbon. So this carbon, first carbon, is known as anomeric carbon. And what is the property of anomeric carbon? That was the part of carbonyl group in the open chain form. So it was the part of the carbonyl group, or you can say in this case, this was the part of the carbonyl group. So this carbon is known as the anomeric carbon, which was linked to double bond O and OH group. So in that way, alpha and beta form are referred to as anomers of each other. So now we'll look into the polysaccharides. So polysaccharide means that there are multiple sugar subunits which are attached to each other. So one such example is of cellulose. So these all are the storage polysaccharides which store the food in the form of, means food is stored in these forms. So cellulose is the means polysaccharide which is present in the plants, and in this case the subunits are constituted of beta glucose. So multiple beta glucose subunits, they are linked to each other by glycosidic bonds, and this linkage is 1,4 linkage, and there are no such branches, and this is the diagram and this is the shape. So this is just a representation how it appears. Then comes starch, which is also present in plants. So starch is composed of amylose as well as amylopectin, and amylose is a alpha glucose. So in the case of cellulose, we have seen there is a beta glucose, whereas in case of starch, we have alpha glucose in both amylose as well as amylopectin. And amylose, we have seen that there is different glucose subunits. They are linked by alpha 1,4 linkage, whereas in case of amylopectin, it is a branched polysaccharide in which multiple glucose are linked by alpha 1,4 linkage, as well as there are also alpha 1,6 linkage. So these are the linkage which are forming the branches. So for the linear chain, there are branches in which the bond is formed from first carbon to sixth carbon, so which is known as alpha 1,6 linkage, and per 20 subunits. So after 20 subunits, you will observe branching. And in case of cellulose, there is no branching. In case of amylose, there is no branching. So in amylopectin, there is branching. And when we talk about the glycogen, which is the animal storage polysaccharide, so it contains alpha glucose and it is also branched. It also contains alpha 1,4 linkage as well as alpha 1,6 linkage. And in this, branching is observed per 10 subunits. So per sub 10 units, you will observe some branching. So in this way, it is a branched polysaccharide. So these are the polysaccharides, and now we'll look into the detailed structure of the starch. Starch is composed of amylose as well as amylopectin. Amylose is present nearly about 10 to 30%, whereas amylopectin is present in about 70 to 90%. So this is the linkage, that how the alpha 1,4 linkage is formed. So as in the previous slide we have seen that alpha sugar, or you can say the alpha glucose, is the one in which the OH group is present away from the means oxygen, which is linked to the anomeric carbon. So in this case, OH is present away from this O group. So that's why it is known as alpha glucose, you can say in this case. So alpha form, and in this way it forms alpha 1,4 linkage, which means first carbon is linked to the fourth carbon of the another glucose subunit. And in this case, you'll see all the glucose units which are present, they are the alpha forms. So they are the alpha glucose. Similarly, if we talk about amylopectin, in this also you can see for the linear chain, there is the alpha 1,4 linkage, whereas for the branching, you will see there is alpha 1,6 linkage. So first carbon of this particular sugar is linked to sixth carbon of the linear chain sugar. So in this way, there is a branching which is observed, and this branch point is present after every 25 to 30 units. Now, next polysaccharide was cellulose, in which there is a beta sugars or beta glucose is present, and it shows beta 1,4 linkage, in which the OH group is present towards the same direction of the O group, or means which is attached to the anomeric carbon. So this is a different representation of you can say beta glucose, and in this case it forms beta 1,4 glycosidic linkage. Along with this, you will also see that there are some hydrogen bonds which are observed between two uh, glucose subunits. So this hydrogen bonding, it leads to the tight packing of these uh, you can say the longer chains of the sugars. So due to this tight packing, you see that they appear as a fibers. So cellulose is appear as a fiber due to this interchain hydrogen bonding. So interchain hydrogen bonding, it closely or tightly packs two sugar units together. So in that way, they appear as a strong fibers, and they show beta 1,4 glycosidic linkage. Now we'll move to fats, but till now, do you have any doubts in carbohydrates about their DL forms or about their alpha beta forms? Because these are the basics, so these have to be cleared. Okay, so let's move to fats, which are our second class of biological macromolecules, and they are composed of triglycerides. So in many textbooks or in many pages, you'll see that they are referring them to as lipids. So they are also known as lipids, and fats are different from fatty acids. Okay, so fatty acids are smaller constituents of the fats only. So lipid is the precise term to call them. Okay, so fats is also means sidewise it is called as fats also. So fats are composed of triglycerides, which consist of three fatty acid molecules, and they are bonded to a glycerol backbone. So when the glycerol backbone is attached to three fatty acids, it constitutes a triglycerides, and it serves as a major energy source, and it plays key role in cell structure as well as signaling. So mostly in signaling, the fats are observed, such as, for example, in our body there are multiple hormones. So all those are composed of fats. Now, in order to see the classification of these lipids, so there are three main categories. One is simple lipids, then compound lipids, and derived lipids. So simple lipids mean the triglycerides, simply the three fatty acids with glycerol backbone. So they comes under simple lipids, in which we see glycerol or the fatty acids. So fatty acids are further divided into saturated fatty acids as well as unsaturated fatty acids. And unsaturated fatty acids could also be divided into monounsaturated or polyunsaturated. So as we know that unsaturated means that there are double bonds, and those double bonds, they leads to bends in the straight chain. So monounsaturated means if there is only a single double bond present. Polyunsaturated means if more than one double bond is present. So that is referred to as polyunsaturated. So this comes under simple lipids. Simple lipids means triglycerides which are composed of glycerol and fatty acids. Then next type of simple lipid is waxes, such as honeycomb or ear wax. So they comes under the simple lipids. Then comes compound lipids. So compound lipid mean that along with lipid, you have some other groups which are attached. So in that way, they form a compound. So phospholipids comes under the compound lipid, in which you will also observe a phosphate group. Then comes glycolipids, in which you will see a means a sugar or carbohydrate group is present in the lipids. So it comes under glycolipids, such as cerebrosides or the gangliosides. So there are multiple, but we won't go into their details. Then comes derived lipids. So derived lipids are the ones which are derived, which are not direct lipids, but they are derived. There are certain other processes which undergoes, and then these lipids are synthesized, such as steroids, sterols, and carotenoids. So in steroids, as we see the bile acids or sex hormones, sterols such as cholesterol and ergosterol, and then carotenoids such as keratin or xanthophylls. So these are the colored pigments which are present in the plants. Okay, so this is the structure of the triglyceride. So it is composed of a glycerol backbone with three fatty acids. So in this case, this is a glycerol backbone, and there are three different fatty acid units which are attached. So in this way, triglyceride is synthesized. Now we'll see means these categories are further elaborated to make you understand that different which backbone is present. So in terms of simple lipids, as we have seen, there is a glycerol backbone with three fatty acids being attached. Then we have seen the compound lipids, in which we studied phospholipids. So in phospholipids, if we talk about the glycerophospholipids, so glycerophospholipid means there is a glycerol backbone, two fatty acids, one phosphate group, and it is attached to a alcohol group. So in that way, it comes under glycerophospholipids because phosphate group is present. Glycero means glycerol backbone is present, and it is lipid because it contains two fatty acids to it. Then comes sphingolipids. So sphingolipid means in this backbone will be a sphingosine. Instead of glycerol, if we have a sphingosine backbone, then it is a sphingolipid. One fatty acid and phosphate group and a choline group is present instead of alcohol. So this comes under phospholipids. Then glycolipids. So if in glycolipids, if we have a sphingolipid, so sphingolipids means it has a sphingosine backbone, but if it comes under glycolipid, that means it will have a mono or oligosaccharide. So sphingolipid is a subcategory depending that on which bigger category it comes. So if it comes under phospholipids, then it will have a phosphate group with choline. If it comes under glycolipid, then it will have a sugar moiety or a carbohydrate group attached to it. Similarly, we have a galactolipids or the sulfolipids. So in this case, we have the glycerol backbone, two fatty acids with monosaccharide because it is a glycolipid, so it should contain a carbohydrate group. And along with this, it also contains a SO4 group. So if it contains a sulfur group, then it comes under sulfolipids. So from the name only, it is clear that what are the constituents of that lipids. And these are the membrane lipids. So they are polar in nature and they are mostly observed in the membrane. Then there are some archaeal ether lipids. So they are present in the archaeal or those primitive organisms. So in them, they have a glycerol backbone with diphytenyl, two diphytenyl rings with phosphate group, and then again another glycerol group, and then phosphate group and another glycerol backbone. So these are the special kind of lipids which was observed in archaeal ether lipids. So as we also know that the archaeal organisms or these are the primitive prokaryotes, so they exist in the extreme environments. So they could even survive the extreme temperatures conditions. So these lipids, due to these linkages, they were able to survive in those temperatures. So these are the archaeal ether lipids, and this is their configuration, or you can say the constituents which are present in these lipids. Now, there are another steroids and waxes which are not included in this. So steroids, there is a difference between the steroids and sterols, and many people get confused in that. So steroid is a bigger category or a bigger term. So steroid means if there are four rings, ring structure. So these are four rings, and if these four rings are present, so they are known as steroids. And steroids are found in the cell membranes where they influence the membrane's fluidity. And that particular steroid is the cholesterol. So cholesterol comes under these steroids, which are helping in the membrane fluidity, and they can also act as hormones in the body. So steroid is a bigger term in which there are other categories which are present, such as sterols, in which the steroids contains the alcohol group. So in that way, it is a subcategory of steroid referred to as sterol. So cholesterol is one of such steroid which contains the alcoholic group. And then comes waxes. So they are the esters made up of alcohol chains and a fatty acid chain. So in this way, and you have also observed that in plants, their leaves, some of the leaves contain the waxes due to which they are means they do not react with or they do not interact with water. So they are non-polar in nature, and they have the means this particular wax helps them to prevent the water loss. So there is a process named as transpiration with which the water is lost from the leaves. So to avoid that, waxes are present, which help them to survive in the means extreme conditions where water is less. So water scarcity in that condition, waxes are helpful. Yeah, so this is a steroid ring, and this is a sterol, which is cholesterol, which comes under the sterol. So in this case, there is a hydroxyl group which is attached to the steroid ring. So that is a subcategory of steroid, which is sterol. Okay, so now I'll move to nucleic acids. So is there everything clear about the lipids? Okay, so yeah, we'll talk about the nucleic acids. So nucleic acids, they are composed of nucleotides, and nucleotides are further composed of nucleoside and a phosphate group. So nucleoside means sugar plus nitrogenous base. So they combine to form a nucleoside, and then they attach to a phosphate group and they forms a nucleotide. So nucleic acids, they are composed of these subunits. And there are two types of nucleic acids, DNA as well as RNA. And depending on what type of sugar is present, this DNA and RNA can be distinguished. So this is a nucleotide and this is a sugar. So in this case, it is a ribose sugar. So if there is OH group present in the second carbon is attached to OH group and H group, then it is the ribose sugar. If this OH group is replaced with hydrogen, then it is referred to as deoxyribose sugar. So that is the main difference among DNA and RNA. And these are the constituents. So in case of RNA, you will see in place of thymine, there is a uracil. And rest other three nitrogenous bases are same in both DNA and RNA. And one another difference is that RNA is a single-stranded structure, whereas DNA is double-stranded, in which these nitrogenous bases are connected to each other. And there is a sugar phosphate backbone. So this backbone is composed of sugars, which are two sugars are attached to each other by a phosphate group, which is referred to as a phosphodiester linkage. So in this case, this backbone is composed of sugar phosphate, and each two sugars are linked to each other by phosphodiester bonds. And in case of DNA, where we have seen the complementary base pairing among the nitrogenous bases, so this complementary base pairing is with the help of hydrogen bonds between two nitrogenous bases. And in this case, there are two purines, that is adenine and guanine. These are referred to as purines. So purines are the nitrogenous bases which contain double ring structure. So if double ring structure is present, they are purines, and single ring structure are referred to as pyrimidines. So for example, cytosine, thymine and uracil, they are the pyrimidines. Adenine will always pair with the thymine. So the purines pair with pyrimidines. So adenine pair with thymine, and in case of RNA, adenine pairs with uracil. So whenever adenine will pair with thymine or uracil, they will form double bond, yeah, two hydrogen bonds. But when guanine and cytosine will base pair with one another, they will form triple hydrogen bonds. So means due to the number of oxygen and hydrogen groups present, they will determine that how many bonds are possible. So in case of guanine cytosine, triple hydrogen bonds are possible. So we won't go into much of the details. Now let's quickly look about the advancement which are made in the nucleic acid structural studies. So Rosalind Franklin was the one who studied the X-ray diffraction pattern for the DNA, and she has given that diffraction pattern in 1952. And later on, Watson and Crick, by observing the diffraction pattern, they have given the double helical structure of the DNA. They said that DNA is arranged in the double helical form. So this is the detailed structure of the polynucleotide, or you can say the DNA, in which this is the sugar phosphate backbone. Each sugar group is attached to nitrogenous base. So these are different nitrogenous bases. And then there is another sugar phosphate backbone which has the another nitrogenous bases. And these two helices, or you can say the two helices run anti-parallel direction in the DNA. So they are linked to each other by forming the hydrogen bond between their nitrogenous bases. So in this case, A and T are forming double hydrogen bonds, whereas C and G are forming triple hydrogen bonds. So this is the detailed structure of a DNA. Okay, so now we'll move to the fourth class of macromolecule, which is the proteins. So these are the large and the complex molecules which are made up of amino acid chains. So in the nucleic acids, the smaller subunits which combine together to form a nucleotide is uh, to form a DNA is the nucleotide. So in this case, in case of proteins, the smaller subunit are the amino acids. So multiple amino acids combine together to form a protein, and they fold into specific three-dimensional structures which are serving the critical functions, such as they can catalyze biological reactions. So enzymes are the type of proteins which are involved in catalyzing certain reactions. So they act as a catalyst which can enhance the processivity of a particular protein. So two proteins are interacting in making a means product. So they catalyze this reaction, and they support the cell structure and regulate biological processes. So there are 20 kinds of R groups which are present, and depending on the R group which is present, the proteins or the amino acids can be of 20 different types. So there are 20 amino acids, and the next means this is the backbone of an amino acid, in which we have the amino group, we have the carboxyl group, we have the alpha carbon which is linked to one hydrogen and one R group. So everything remains constant, only R group changes in different types of amino acids, and this is known as the alpha carbon. Now the linkage which is present between two different amino acids, that is referred to as a peptide bond. So in DNA we have seen the phosphodiester linkage among sugar phosphate and hydrogen bonding among the nitrogenous bases. In sugars we have seen that two different sugar units are linked to each other by glycosidic bond. So similarly, in case of proteins, the bond which is present among two amino acids is the peptide bond, which is a covalent bond in nature. So it is a CONH bond. So in some textbook it is also written as a CONH bond, in which the carboxyl group of one amino acid is linked to amino group of the another amino acid. And in proteins you will always see that it is from N terminal to C terminal. So the first amino acids N group will be exposed, whereas the terminal amino acids C group will be exposed. So in that way, proteins are always read from N to C terminal, and these are different amino acids, and depending on the R group there are different types of amino acids. For example, they could be non-polar or aliphatic R groups where there is no charge, and these non-polars they cannot dissolve in water, so such as glycine, alanine, valine, isoleucine, methionine, proline, leucine. So these are the non-polar aliphatic amino acids. Then comes polar or uncharged R groups. So these are uncharged, but they are polar in nature, such as cysteine, serine, threonine, asparagine, glutamine. So these come under polar uncharged amino acids, whereas there are certain amino acids which are positively charged, such as lysine, arginine, and histidine. And then there are certain amino acids which are negatively charged, such as aspartate and glutamate. And then there are certain aromatic amino acids which contain an aromatic ring, such as phenylalanine, tyrosine, and tryptophan. So, on the basis of the R group, these are the categorization of 20 different amino acids. And what are the functions of the proteins? So, they can have enzymatic, there could be enzymatic proteins such as pepsin, which can help to catalyze the reactions. These can also help in defense, such as antibodies, which are helping in defense. Then storage proteins such as ova albumin, and then there are certain proteins which are also helpful in transportation. So for example, hemoglobin, which carries oxygen in the blood, so that is acting as a transport protein because it is carrying oxygen. So these are different functions of the proteins. And if we talk about the protein structure, then there is a primary structure, which means if you only read the linear sequence of amino acids, so that is referred to as primary sequence. If you consider the hydrogen bonding among these peptides, so they are referred to as secondary structure, in which there could be alpha helices or beta sheets, so which are observed as a pleated sheets, so they are beta sheets. So in this case, when you are observing the hydrogen bonding among these peptides, so peptides are the smaller amino acids or smaller group of amino acids which are present in the protein, so that are known as peptides. So if there are those hydrogen bondings which are present in those peptides, then they form the secondary structure. And then tertiary structure is the one when you observe the three-dimensional folding of the protein, in which along with hydrogen bonding, you are also observing the other known covalent interactions, which could be van der Waal interactions or other side chain interactions. So when they come into picture, you will be able to see the protein tertiary structure, which is the folded structure. Then comes quaternary structure. So protein can have multiple chains, or you can say protein can be constituted of polypeptides. So which means if multiple chains are present and they are folded onto each other, so that is referred to as a quaternary protein structure. And these interactions, the interactions which determine the tertiary structure of protein includes hydrogen bonds, and the hydrogen bonds could be between the side chain and the carbonyl group of the group on the backbone, or it could also be present between the two side chains. So these interactions also determine what could be the tertiary structure. Along with this, there are some hydrophobic or van der Waal interactions which are also playing a role. Then disulfide bond is also one of the interaction. So these sulfur groups are present in the cysteine amino acid and as well as in methionine also, but mostly cysteine is involved in forming the disulfide bonds. So cysteine is present. So in that case, you will also see that the amino acid will bond to each other. So in that way, it will determine that how the protein will fold itself. Then comes ionic bonds. So there are ionic bonds also which are present. So in this way, these bonds will determine, these interactions will determine what could be the fold of a protein. So that's why the primary sequence of a protein is very important to determine its structure. So if you change the amino acid, it might influence the structural fold which it could take. Then tertiary structures, they are diverse. They are composed of, if they are mostly composed of alpha helices, they could also be composed of most of the beta sheets, or there could be a combination of uh, alpha helices and the beta sheets, or they might have multiple disulfide bonds. So depending on the structure, they could be diverse. There could be diverse protein structures possible. Now we'll talk about the three secrets of covalent bonds. So the 3C, which is present in the covalent bonds, which determines its configuration, conformation, and chirality. So why these things are important to study, what configuration means, so that we will see now. So when we say configuration, it means the fixed spatial arrangement of atoms. So whenever we study any protein or any amino acid, so when we look towards its fixed spatial arrangement, that is referred to as its configuration. That means for a particular amino acid, its configuration is always fixed. It will not change until we break any bond. So to change the conformation, we need to break the bond, and the configuration determines what is the reactivity of a molecule and its bonding type. So how it is bonded and how it will interact with other molecules. So this configuration is very important to determine. For example, L amino acids and D amino acids. So these are two different configuration of amino acids. So now, as in the right side, as you can see, so these are two representation or two different configurations, and these two configurations, they are the mirror images of each other, in which the central C, or we can say the chiral carbon or the central carbon, or we can say alpha carbon. So in this amino acids, the carbon which is linked to the amino group and the carboxyl group is referred to as the alpha carbon. So if the alpha carbon contains the NH2 group towards its left, so that is L amino acid, and if that NH2 group is present towards the right, it is the D amino acid. And this particular representation is known as Fischer projection. So it is similar to that of, as we have seen in carbohydrates, the L form of sugar or the D form of sugar, but the difference is that in that case we were looking at the OH group, But in this case, we will be focusing on the amino group. So this is the difference among the L and D forming the sugars versus L and D forming the amino acids. When the NH2 group is kept as a reference and its position determines the type of amino acid. Then there are later on another scientist came who has given the R and S forms. So L and D form is only giving you the idea of the relative configuration. It does not tell you about the absolute conformation of a particular amino acid. So later on, R and S forms came up. So in this case, amino acid is drawn in the form of sp3 hybridization, so which adopts a tetrahedral structure. So in this case, what is being done to identify what could be the configuration? So in this case, there is a means on the basis of priority, we perform ranking of the atoms, or you can say ranking of the molecules which are present in the amino acid. So in this case, so in the next slide, we'll see that how this ranking is done. So based on that ranking, we'll look for the directionality of its movement. So if its direction is anti-clockwise, then it is the S form. If that directionality after giving the ranks, if it is the clockwise, then it is known as R configuration. So in the next slide, we'll see that what is the normal configuration which is generally observed for all the amino acids and how it is determined. So next thing comes the conformation. So after configuration, the conformation. So conformation means different spatial arrangement of atoms that can be achieved by rotating around the single bonds. So in this case, if we want to change the conformation of a protein, we do not have to break any bond as we had to break in configuration. So in conformation, we simply have to means rotate it around the single bond. So in that case, multiple conformations can be adopted. Then comes chirality. So chirality means the carbon, when it is attached to four different subunits, it is known as the chiral carbon. So in that way, chirality means a property of a molecule in which its mirror images are non-superimposable. That means they cannot be perfectly aligned. So a molecule is chiral if it has at least one chiral center. So if you constitute a bigger molecule and it contains a single chiral carbon, then also it is referred to as a chiral. Whereas if there is no chiral center, then it is achiral. So in case of amino acids, the amino acid which is known as glycine, so it contains, in place of R group, it contains hydrogen. So in this case, there is no chiral carbon which is present. So in that case, glycine is a achiral, whereas rest other amino acids are chiral in nature. Yeah, so now we'll just quickly see to clear the concept that what the S and R form means. So firstly, if we talk about L and D forms, so it is on the basis of NH2 group. If NH2 is present on the left, it is the L form. If it is present on the right, it is the D form. And it has been known that all the 19 chiral proteinogenic amino acids, they have L configuration. So L configuration is the one which is widely observed in nature. And the exception is L cysteine. So means out of these 18, means out of the 19 amino acids which are having the L conformation, uh, means 18 have the S form, whereas the L cysteine, it has the exception, it shows R form. So for example, if we have L-valine, so in this case, the NH2 group is present, means in all these cases, in all the L forms, the NH2 group will be present on the left sides, and these are the other functional groups which are present. So now I'll show you that why these functional groups are important. So as we discussed in the previous slide, that there is a naming, there is a priority which is done, and with that will determine in which direction a molecule can move, whether it will be clockwise or it will be anti-clockwise. So that particular priority is depend on the type of atom which is present. So if oxygen is present, so it is given the higher priority. And before oxygen, if sulfur is present, so it depends on the periodic table. What is the position of that particular element in the periodic table? So highest priority is given to sulfur, then to oxygen, nitrogen, carbon, and least priority is given to hydrogen. So what you have to do is you have to take a amino acid and you have to start its ranking starting from nitrogen. So in this case, nitrogen will always be given as rank 1. The next priority is given to the element which occurs in the second position. So in this case, for example, let's consider the case of L-serine. So in this case, first priority is given to nitrogen and H2 group because it lies at the first position. Then second priority is given to the from carbon and hydrogen, the priority will be given to carbon. Now there are two carbons. So how to decide to which carbon we should give priority? So now we will consider the second group or the second element which is attached to this carbon. So in this case, it is oxygen, whereas in the other case, it is hydrogen. So the priority will be given to the carboxyl group. So in this case, second priority will be given to C double bond OH, and then hydrogen will be given the fourth priority, whereas CH2OH will be given third priority. So in this case, the L-serine will show the anti-clockwise or the counterclockwise rotation. So it will be S-serine. Whereas in case of L-cysteine, if we have to rank it, so in this case, what happens is that the second priority is given to its R group, which is CH2SH, because sulfur is present. So when we are going to decide the priority, we'll see the carbon is constant in both, but the next group which is attached, in this case, it is double bond O. So among OH and double bond O, the priority is given to the one which contains double bond O. So in the previous case, although it has CH2 and OH group, but priority is given to double bond O group. So that's why we selected COOH as second group. So in this case, the carbon is attached to sulfur. So in this case, the priority is given to CH2SH instead of C double bond OH. So now NH2 will be given first priority, CH2SH will be given second priority, and then C double bond OH third priority, and hydrogen fifth priority. So it will show a clockwise rotation. So in that way, it comes under the R configuration. So in this way, all the L amino acids, which are means in nature they exist in the L form, so they are always S. They shows S configuration, which is anti-clockwise or counterclockwise, whereas L-cysteine is an exception, which shows R configuration. And another thing to remember this is that whenever the R group has a lesser priority than the C double bond OH group, then it will always have S configuration, and only in the rare scenarios where it shows the higher priority than the C double bond OH group, then only it will show the R configuration. So this is all about the S and R form, and this is just for your clarification. I don't think this is important from the exam point of view. So is this clear? Because this was a bit complicated. So is it clear till now? R and S forms? I think, okay, so yeah, thank you. So maybe when you will go through these slides again, then you'll be better able to understand. Okay, so it's already 7:20, so I'll quickly go through the content so that I don't waste much time. So what is the importance of these three C? So they are helpful to understand the shape of the biological molecules because they determine what would be the folding of particular protein. With that, they also determine the stability. Along with this, they are also helpful in determining what could be the recognition of that particular molecule, how it will bind with another molecules. So these configurations help you to decide this. So that's why they're important. That's why it's important to study the configuration of a protein or its conformation. And it's important to know that whether it contains a chiral carbon, whether it is a chiral or achiral, because it determines that how it will interact, how it will fold. And with particular type of folding or particular type of interactions, it could vary in its functionality. So in that way, it is important to study these three C's. Now we'll talk about the central dogma of life. So this theory was given by Francis Crick in 1958, and he said that information flows only in the one direction. That means information flows from DNA to RNA, and in formation of DNA to RNA, that is known as transcription, when DNA is converted to RNA, and ultimately the RNA transfer this information to the proteins, so which is known as translation. So this is flow of information unidirectional from DNA to proteins. And he said that there is means this information cannot flow backward. Whereas they also know that RNA can convert to DNA by reverse transcription. So this was already known. But protein can never give means can never produce DNA or in that way. So it is only in one direction. But later on, different scientists performed discoveries, and then prions came, and prions shows exception to the central dogma theory. So prions are the infectious proteins which replicate without going through DNA or RNA intermediates. So in that way, they do not have to, means they do not require DNA to form proteins. They are themselves proteins which replicate, which perform multiple copies of themselves. So in that way, it somewhere it is means it negatively enhance means negatively it does not support the central dogma of molecular biology given by Francis Crick. So it does not support it. And then this is the codon table. So in this way, I wanted to show that how a nucleic acid can decide the protein sequence. So whenever a nucleic acid converts to RNA, then it forms a single stranded RNA molecule which can contain multiple nucleotides. So each three nucleotides together constitutes a codon on the mRNA. So mRNA can get means translated to protein. There are multiple RNAs which can be formed, in which mRNA is the one which is known as messenger RNA, which translates and forms protein. So in that mRNA, the nucleotide sequences which are present, they decides that what could be the possible amino acid it will give rise to. So in that way, if you know the sequence of RNA, you can predict the sequence of the protein. So this is the codon table which tells you that what could be the possible amino acids which a nucleotide sequence can form. So in this way, the information is linked. Now this is the composition table of biological macromolecules which we have seen so far. So in case of proteins, there is carbon, hydrogen, nitrogen, oxygen as well as sulfur. In nucleic acids, we have seen carbon, hydrogen, oxygen along with phosphorus and nitrogen. In carbohydrates, we see carbon, hydrogen, oxygen, and in certain cases, there is also nitrogen which is present. And in lipids, there is carbon, hydrogen, and oxygen. So these are the atoms which are present. And then there are building blocks. So proteins are composed of amino acids. In nucleic acids, we have seen nucleotides are the building blocks. In carbohydrates, there are simple sugars. And in lipids, fatty acids as well as glycerol combine together to form the lipids. Now, how they bond to one another. So in proteins, we observe peptide bonds. In nucleic acids, we have seen phosphodiester bonds. In carbohydrates, the linkage is glycosidic. And in lipids, they are ester bonds and they form various di and triglycerides. And then these are certain interactions, means the bonding interactions which take place. So in protein, we have seen covalent bonding, such as disulfide linkages, they are the covalent bonds, as well as peptide bonds, they are also the covalent bonds only. Then hydrogen bonds, charge-based interactions, which are ionic interactions, and then van der Waal interactions, which are also taking place. So these are between intramolecular interactions, which can take place inside means within a single molecule. So these are intramolecular interactions. In nucleic acids, we have seen hydrogen bonding between two nitrogenous bases as well as stacking interactions. In carbohydrates, there are only hydrogen bonds which are present. So in case of I think cellulose, we have seen that how the hydrogen bonds they help them to make a fiber-like structure. So there are many intramolecular hydrogen bonding which is present. In case of lipids, there are van der Waal bonding. And then about the functional roles. So proteins can act as enzymes. They can act as structural proteins. They can also act as sensors as well as receptors. So if they are present on the membrane, they act as the receptors with which they bind to another proteins. And then nucleic acids, so they act as the storage of genetic information because they pass on this information to another cells. And then as enzymes and ribosomes. So in ribosomes, they also contain the smaller nucleic acids such as small nuclear RNAs. So in that way, ribosomes are also means due to which ribosomes are formed. So along with protein, there are also smaller RNA molecules which form these ribosomes. So that's why nucleic acids are also playing a role in the formation of ribosomes. And then carbohydrates, so they are helping in storage of energy, structural recognition, and interactions. So in terms of recognition, if we talk about different blood groups that are present, so this blood groups, they vary on the type of sugar moiety which is present. So in that way, carbohydrates help in recognition. Then membrane lipids, lipids are involved in the membranes, signaling energy storage, as well as small molecules are also acting as the hormones. Okay, so this is all about all the macromolecules. Now we'll talk about the protein sequencing and uh, what is the need for this protein sequencing. Okay, so there are three different methods for sequencing the protein. Protein sequencing means to understand the what amino acids are present and in which order they are present. So mass spectrometry and Edman degradation. So mass spectrometry is the one which is widely used for the protein uh, sequencing, whereas Edman degradation is also used. So in Edman degradation, it is means helpful if we only want to recognize the N terminus of the proteins because it is a slower process, you can say. In this, it involves the PITC, which is the chemical compound which attaches to the first or the terminal N terminal amino acid, and then it get released. So by changing the pH, it get released, and then firstly it will take one terminal amino acid. Then we have to perform the analysis. We have to perform liquid chromatography and separation, and another peptide chain which is left after this attachment of PITC with the first amino acid. So this is again recycled and again used with another PITC. So the PITC which attaches to the first amino acid, it is further undergone liquid chromatography, and later on some another analysis is performed to understand that what amino acid it binds to. So with that, we will be able to perform this sequencing, but this sequencing will always start from the N terminal of the protein, and there are many limitations to it. Then if we talk about the peptide mapping, which is done by the help of mass spectrometry. So in mass spectrometry, what we have to do is we take a purified protein, we break it into smaller pieces or smaller peptides by using certain enzymes, which could be trypsin, lysine, C, or chymotrypsin. So there are certain enzymes which digest the proteins into smaller peptides. And then after that peptide mixtures are obtained, we purify them using HPLC, so liquid chromatography is used. Now targeted peptides are further passed on to the mass spectrometers. So mass spectrometry is based on the principle that it will break those peptides into smaller fragment ions. So smaller ions will be formed and those ions will be recorded. So in mass spectrometry, we have different units. So they will detect the ions which are formed from that particular peptide, and those ions will be plotted in the mass to charge ratio versus their intensities. And basis on the mass to charge, we predict the sequence of the protein. So in mass spectrometry, it is based on the smaller peptide sequencing. And then comes de novo sequencing. So when we are mapping the peptides, that means when we are mapping the ions to the peptide sequence or the amino acid sequence, then we need some database which already have the information of those peptide sequences. And then we can check that, okay, this particular mass is obtained from this amino acid and these peptides makes this particular mass. So we need some reference to identify it. So in case of peptide mapping, we have that particular database with us in which we know that which mass to charge ratio correspond to which particular amino acid or which particular peptide. But if we do not have that information, for example, if there is some novel protein which is present or some novel combination, or if there is some mutation which alters the protein sequence, so in that way we need to undergo de novo sequencing in which we do not have database in our hand. So in that way we perform certain calculations depending on the what elements. For example, carbon has particular molecular mass. So in that way, we add on those masses, we perform certain calculations. So there are already some softwares which are available. So in de novo sequencing, instead of using some database to experimentally verify the ions with the sequence, we perform certain calculations and we predict that what could be the possible sequence which can arise from particular mass to charge ratio plot. So these are two different methods for protein sequencing, but still it is a bigger of a challenge. So in Edman degradation, this is the process in which we have a polypeptide. We take phenyl isocyanate. This is a chemical which is which is cyclic in nature. Whenever it interacts with the N-terminal amino acid, it makes the pentose ring-like structure. So it converts into a pentose ring, and then it cleaves it off from the other polypeptide backbone. And then this particular means PITC molecule, which is attached to first amino acid, it is further in the low pH, it is obtained, it is purified, and then that particular amino terminal residue is identified by performing later analysis. So other experiments are performed in which comparisons are made. So in this, they perform HPLC, and after HPLC, the comparison is done with the reference uh, plots. So reference is already available, and then we compare them that where the band is obtained. So on the basis of that, we will be able to identify the amino acid which is present towards the N terminal. So this is quite a time consuming task, and it is best for the short peptides. And if you only want to analyze the N terminal sequence, then it is helpful, but there are certain limitations such as N terminal modifications. So if there is any modification in the N terminal, then it won't be able to make that intermediate product, which is pentose ring. So in that way, it won't be able to determine what is the amino acid. Second is if there is known alpha amino acid. So alpha amino acid is the one in which the amino group is one carbon away from this carboxyl group. But if there is non-alpha amino acid such as isoaspartic acid, so this is the aspartic acid normal and this is the isoaspartic acid. So in this way, the carboxyl group is separated from this NH2 group by two carbons. So in this way, this is not the alpha amino acid. So in such scenarios, it won't be able to make that intermediate pentose ring-like structure. So in these amino acids also, if they are present, we won't be able to perform the sequencing. Then if there are disulfide bridges, then also it won't be able to determine the uh, amino acid sequence which is present. And it also depends on the protein concentrations. So if protein concentration is low, then its result might not be accurate. So it requires high concentration of proteins. And then if we talk about mass spectrometry, so this is the entire process that we take proteins, break into peptides, perform liquid chromatography, and then pass it through mass spectrometer. And whenever it passed through mass spectrometer, that peptide sequence will also be broken into smaller fragments. And then after that smaller fragments are obtained, we pass it through another round of mass spectrometry. So in many textbooks, you might observe as tandem mass spectrometry or MSMS. So that means if there are two mass spectrometry experiments are going at the same time. So in this case, after first round of mass spectrometry, the second round of fragmentation it undergoes. So that is tandem mass spectrometry. So in this, those fragments, firstly, one of the peptide or one of the fragment is selected, and then that is further broken into smaller fragments. And then there are some ions which are formed. So depending on the type of ion, B and Y ions are formed. And then later on, after observing the mass to charge ratio with respect to their intensities, we match it with the already available database and we predict the sequence of amino acids. So there are two types, protein database search and de novo. So de novo comes when we do not have the protein database which contain that sequence. So in that way, we undergo the de novo peptide sequencing. Yeah, so we won't go into more details. So this is already a detailed one. Okay, so now if we here we discussed about protein databases. So what are the protein database which contain this protein sequence information? So first one is protein information resource, which contain the experimental protein sequence informations. And then comes Tremble, which is translated EMBL. So in this case, we have the RNA sequence and we convert or we predict that what could be the possible protein sequence. So this is based on the predictions. Then comes Swiss Institute of Bioinformatics. So this is also based on the experimental plus manual annotation work. So when Swiss Prot and EMBL or translated EMBL and PIR are combined, they form a universal protein consortium, which is UniProt. So in UniProt, we contain the protein sequences from all three different sources, which includes both experimental as well as predicted protein sequences, and they form UniProt database. So it is a protein sequence database. So in this way, these databases are helpful to know that what are the sequences of particular proteins. Okay, so now next thing is DNA sequencing. So after protein sequencing, the DNA sequencing come into picture because DNA sequencing is much could be easily achieved, and this is also means previously it was time consuming, but with the advancement in technologies, now we are able to quickly sequence entire genome as well, with which we can also determine the genes which are now undergoing mutations. Because as we have seen from central dogma, DNA is the key player which will decide that what will be the information which will be passed on to proteins. Although proteins are important, but DNA is the one which is sitting in the back end and which is deciding. So if we can kind of mutate or means change the DNA, then we can also govern that what protein it will design. So in that way, DNA sequencing is also important. So DNA sequencing means when we are sequencing the nucleotides which are present in the DNA. So advantages is similarly because to understand the genomics, molecular biology, medicine, and performing evolutionary studies. So with the help of DNA sequencing, all these things can be done. And it also helps us to understand the genetic makeup of organisms. For example, the primitive organisms such as prokaryotes, which were present in the which were thermophiles and present in the thermal vents. So we can take their DNA and we can also check that what are the genes which are present, that how they were able to survive in those extreme conditions. So in that way, DNA sequencing can help us to understand their genetic makeup and uh, to understand what genes are responsible for certain phenotypes. Then, with the help of DNA sequencing, we can also sequence the entire genome of an organism. So with that, we can also compare among the genome of two individuals. For example, a healthy individual and a diseased individual. So in that way, we can also diagnose the faulty genes, or we can predict, or means we can perform prognostic studies where we can find out that whether this means what are the possibilities that this organism is suffering from which kind of disease, or all those predictions can be done. Then comes exome sequencing, in which instead of sequencing the entire genome, we only sequence the region which codes for protein. So, which means only exonic region, which is the protein coding and not the non-coding regions. So, this is the targeted approach in which we are only performing sequencing of the targeted regions or smaller regions. So, in that way, it is quite fast and less expensive than the genome sequencing. But the limitation is that it might lack, means those known coding regions can also have some regulatory functionality, so we will miss out on those informations. So before moving on to DNA sequencing, we must understand that how this DNA is synthesized. So this process is known as replication, which decides that how the DNA synthesis will take place. And this DNA replication, it is mostly observed in the S phase of cell cycle, where the DNA duplicates. So this is also known as DNA duplication, and it is semi-conservative in nature, which means that one strand is the parent strand and another strand is the daughter strand, which is the new strand. So it is somehow conservative. It is conserving one of the parent strands. So that's why it is referred to as semi-conservative, because one strand is parental and one is new daughter strand. And now let's quickly talk about different enzymes which are involved. So first one is helicase, which is responsible for unzipping the DNA. So DNA is double-stranded. There is complementary base pairing, and those helices are helically arranged. So in that way, we need to unzip the DNA so that we can start its replication or forming its uh, duplicate daughter strand. So in that way, to unzip it or to break those hydrogen bonds, we need helicases. And after unzipping it, the DNA has the ability that it tries to re-anneal, it tries to bind back. So to avoid that re-annealing, we use SSBs, which are single-stranded binding proteins. So these single-stranded binding proteins, they bind to that unzipped region and they make sure that this single-stranded DNA do not anneal, do not move back into the double-stranded form. So for that stability, it is needed, as well as it also protect the single-stranded DNA from nucleases damage. So there are certain enzymes which are known as nucleases, which break the nucleic acids. So to avoid that breaking, single strand binding proteins are important. Then comes topoisomerase. So whenever this unzipping is happening, so in that way, the on the opposite end, there is some supercoiling which also takes place, because when you are unzipping it, so there is some stress which is taking place on the opposite end of that unzipping region. So to avoid that supercoiling, we need another enzyme which is topoisomerase, which makes sure that DNA is not under stress. Because if you will tightly unzip it, then it might break from the ends. So topoisomerase is responsible for that. Then comes primase. So as if you might have read in your molecular biology class that DNA polymerase cannot initiate the process of replication, so it requires some RNA primers. So these primers act as a starting point from where this initiation can take place, from where the new DNTPs can come and they bind to that particular RNA primer. So primase have that role. And why we need primase in case of DNA? Because another thing is that in this case, in the sugars, we have deoxyribose sugar, which do not have OH group at the second position of the DNAs, means the sugar which is present in the second position. So do not have OH group, due to which we cannot make that bond. So to avoid that, we need primase, which constitutes a RNA primer and which has that OH group. So due to which we can further start on the elongation of particular nucleotides. And then comes DNA polymerase, which is responsible for binding the DNTPs to the primer. And then RNase H, which will remove the RNA primers in the end after the elongation has happened, and then it will constitutes the DNA to, means it attaches another nucleotides in the place of that primer, and then ultimately DNA ligase will seal those gaps which will come in between. Yeah, so this is the entire machinery of prokaryotic as well as eukaryotic DNA replication. So I will just quickly go through the prokaryotic one. So in this case, helicase is unzipping this DNA, and where the helicase will start unzipping, so it is the origin site. So this is a simple, or you can say this is a linear diagram, but in case of prokaryotes, we have the circular DNA and which is double stranded. So in that there are certain regions which are known as origins. So origin are the sites from where the replication begins, and these ori sites, they are rich in AT bonds because AT are the double bonded structures. So it is easier to break double bonds rather than CG, which are the triple bonded groups. So in this way, helicase will start that unwinding from the AT rich region, and it is now moving forward. It is unwinding the double stranded DNA, and now the another enzymes are also playing their role, such as topoisomerase is maintaining that there is no stress or no supercoiling on the forward part. And then after the helicase, the DNA polymerase, or for example the primase is coming, it is synthesizing a primer, and then DNA polymerase is coming and it is attaching the nucleotides to the primer. And one side of the DNA strand is the leading strand, because DNA always synthesizes from 5 prime to 3 prime direction, but towards the other strand where it is running from 5 prime to 3 prime, so in that way synthesis cannot start from 3 prime. So in that way there are shorter fragments which are synthesized from 5 prime to 3 prime, and they are referred to as Okazaki fragments, and this strand is referred to as lagging strand because it is having shorter fragments instead of the single continuous stretch. So that's why it is known as lagging strand. Yeah, so ultimately, after all the synthesis, the another enzyme will come which will remove these primers which are attached, and it will make a continuous double, means DNA strand only. It will make sure that there is no RNA present. So that's why we need to remove the RNA primers, and the gaps which are remain, they are sealed by the DNA ligase. So it will seal the gaps between the two DNTPs which are attached. Yeah, so in eukaryotic DNA replication machinery, it varies only in the type of enzymes which are involved, because there is means eukaryotic machinery is quite complex. So there are different enzymes which are used. So we won't go into their details. If you want to study, you can read from these figures. And then before moving on to DNA sequencing, we also need to understand about the PCR, which is the polymerase chain reaction. Because if we want to perform DNA sequencing, we also need a reasonable sample to do it. So as you have seen, the forensic experts or different people, when they collect the sample, that sample is very minute. So we need to amplify it. We need to make multiple copies out of it. So for that, we perform PCR, which is polymerase chain reaction, in which we amplify a specific DNA sequence, which means we make multiple copies of that single DNA, so that whenever we go for performing sequencing, then we have enough sample with us. So it was given by Kerry Mullis in 1983, for which he also won the Nobel Prize. And this PCR, it involves three main steps. First is denaturation, annealing, and extension. So in denaturation, we separate two DNA strands. So as we have seen in replication, that we need helicase to unwind the DNA. So similarly, in when we are performing it in the lab, so we denature the double-stranded DNA. We perform annealing. So in annealing, we allow the double-stranded DNA, which is converted to single strand, we allow the primers to bind to it, so which is known as annealing, and we change the temperature. So for denaturation, we increase the temperature so that the hydrogen bonds break, and for annealing, we try to reduce that temperature now. So we slowly reduce it from 95 to about 55, so that now RNA primers can attach to it, and as soon as RNA primers attach, we use the polymerase which will perform extension. So in this case, the polymerase which is used is the Taq polymerase, which is obtained from the organism Thermus aquaticus, which can tolerate the higher temperatures. So in this case, to avoid the annealing of two single-stranded DNA, we use higher temperature conditions so that the two parent strands, they remain far away from each other. So in this case, as temperature is high, so we cannot use DNA polymerase because it is not stable at that higher temperature, whereas Taq polymerase is stable. So in the PCR, we use Taq polymerase. It will perform extension, it will attach the DNTPs, it will bring the DNTPs and attach it to the primer, synthesize it, and similar enzymes are used, such as ligases will be used, topoisomerases will be used. So other enzymes are also used as such, which are not mentioned here, and buffers and all those things are used so that it mimics the cellular environment. So in this way, PCR help us to amplify the smaller sequence of DNA to make multiple copies of that. And then we move on to DNA sequencing techniques. So there are multiple, as advancement has happened. So there are three main generations of DNA sequencing techniques. First generation was one where Sanger sequencing was used, Maxim Gilbert. So these comes under first generation sequencing. Then after some advancements, second generation sequencing came into picture. So after first generation, all the sequencing techniques, they are also known as next generation sequencing, such as Iron Torrent, Illumina, and then currently there are means long read sequencing, in which at the same time only you can sequence the entire genome instead of breaking the genes, or instead of sequencing smaller genes, we can sequence entire genome at the one go. So they are known as long read sequencing. So which comes under third generation, such as Pacific Biosciences, which is PacBio, and Oxford Nanopore. So means with the advent, we are also increasing the size of the DNA, means what could be the possible base pairs which can be sequenced in the one go. So with this, we have moved from short read sequencing to the long read. Now let's quickly look about the what the Sanger sequencing means. So Sanger sequencing is also known as chain termination method. So what chain termination means? That instead of using normal DNTPs, we use DDNTPs, which are di-deoxynucleotides. So in this case, as we know in deoxy sugar, we have H group in the second carbon. We do not have OH group. But in di-deoxy, in third carbon also we have the hydrogen. So we loses OH group at third carbon also. So in this way, if there is no OH group present in the third carbon, so it will not be able to form the phosphodiester bond. So it won't be able to bind to another sugar with the phosphodiester bond. So in that way, it will not help to elongate the DNA molecules. So it will stop the elongation of DNA molecules. So in this way, Sanger sequencing is based on this concept of chain termination. So in this way, uh, four different reactions will be run. So in that, different types of DDNTPs will be used. So for example, in one mixture, we will use only DDATP. In another mixture, we only use DDTTP. So in that way, wherever thymine is present, so in that way, DDATP will bind and it will stop the elongation. It will not allow it to further elongate. So multiple different fragments will be obtained. Later on, analysis will be performed depending on what was the means previous DDNTP which was present. So these DDNTPs, to distinguish them, they will be differently labeled with different fluorescent dyes. So with that, we can track that what was the position of a particular nucleotide in the DNA. So this is the primitive or the most, you can also call it as a gold standard. So this was the first DNA sequencing method given by Sanger based on chain termination. And the only limitation is that because in this case it is terminating the DNA synthesis and it is also using different fluorophores. So that's why it is not much used. And another problem is that it is only for the short read sequencing. So if the means if you have a smaller genome size, then it is helpful, or if you have a smaller fragment of DNA, then it is helpful. But if there is larger fragment, then the problem can arise. But it is accurate, so in that way, it also have certain good points as well. And if we talk about next generation sequencing, then in this case, the workflow is that we obtain the sample and we generate a sequencing library. We perform target enrichment, we perform sequencing. So in the target enrichment, for example, if we only want to sequence a particular region, so that is known as target enrichment, that we can select that which part we want to perform sequencing. And then after performing sequencing, we perform analysis. So in that way, this analysis is based on the means there are certain computational methods. So past few sequences are obtained, and then there is some reference sequences which are already available. So we map those obtained sequences with the reference to assemble it and make a single larger stretch of DNA, and then interpretation is done. So this comes under next generation sequencing, and this will be much clearer in the later slides at what I was saying. Okay, so if we compare among these different generations, so with each new generation, we are increasing the size of the genome, which we can base pair in a single, so number of base pairs which we can sequence in a single run. So with each generation, the number of base pairs which could be sequenced, they are increasing, as well as cost is decreasing. Yeah, so this is a graph which is showing that means data output per run. So what was the data output? So with the advancement, the output is also increasing for the short read as well as for the long read also. There is increase in the output which was generated. And then this is a comparison among Sanger sequencing and NGS, which is showing that if we have the sample size less than 20, then Sanger sequencing is best because it has the higher sensitivity and it is quite fast. Whereas if the sample size is more than 20 samples, then we should go for NGS. And then comes whole genome sequencing. So in whole genome sequencing, what we do is we break the gene into smaller fragments and then we separately sequence them using different NGS techniques. And after performing the sequencing, multiple reads will be obtained and those reads are assembled onto each other. So in this case, we have a reference genome. We take different reads and we place them to the region to which it aligns. So alignment means where the sequence similarity is present. So in that way, we can join them in the end and then we will be able to know that what are the sequences which are present in the sample. So for example, in this case, reference genome and individual genome is compared. So this is generally done. For example, if a person is diseased, then we can compare his genome with the reference genome. So this comes under whole genome sequencing, and this means uh, we are obtaining the short reads, we are assembling them by placing them on each other, means depending on their sequence similarity, and then we will overlap them and compare it with reference so that we can see where the differences are there. So this will help us for the disease diagnostics or mutations, or we can also check for the single nucleotide variations or small single nucleotide polymorphisms. So these all things can be done with the whole genome sequencing. Then Human Genome Project came in which entire human genome was sequenced. It began in 1990s and it was completed by 2003. And then if you want to know that what were the genes, which means what were the organisms from where the DNA was collected and then sequencing was done. So according to the this particular site, they have mentioned that means this is the main site of Human Genome Project. So in this they mentioned that they have taken means one individual of blended ancestry and 19 individuals mostly from European ancestry. So this percentage was 70% of this blended ancestry and 30% of European ancestry. So different means individual populations were considered to generate a single human genome project in which entire human genome was sequenced. So one single organism cannot be used. So it is mostly done by taking multiple individuals and sequencing their entire genomes and then comparing among their genomes. So after that, reference human genome sequence was generated. So this was it about Human Genome Project, and its aim was to sequence the whole genome. So our genome contains about 3 billion base pairs, and then create the physical map of genes, that where the genes are arranged in the human genome, and to identify disease-causing genes, as well as to understand the function of genes, and to make this information available to researchers and to further develop certain tools for further analytical studies. So this was the aim of Human Genome Project. And after the Human Genome Project was over, when we obtained the sequence, so next thing was that how to go for further analysis, so which comes under post-genomic era, which included the transcriptomics, proteomics, metabolomics, because now we have the genome. Genome will decide what will be the transcriptome. So transcriptome means what will be the RNA which it will form. Then after the RNA is synthesized, it will determine what will be the protein it will form. So which comes under proteomes. And then proteomes, different proteins will interact with each other. They will perform certain reactions, and they will give rise to metabolome. So metabolites, so they will make, this study of metabolite comes under metabolomics. So in this way, in the post-genomic era, the shift has been seen from the genome towards the metabolome. So why it is important? Because the genotype governs the phenotype of an individual. So this comes under post-genomic era, where different sciences came into picture, different studies came into picture. And then there are certain databases which are also needed because we have a lot of information, so we need to document it. So these are some of the example of databases where the information is stored. And then the last part is about protein structure determination, because now we have sequenced the DNA. So it's tricky to identify the structure of DNA. So we just look for the structure of protein, because different protein-protein interaction is governed by the structure of protein. So there are different methods for protein structure determination, such as X-ray crystallography, NMR, cryo-electron microscopy, and they vary in the method which they use. So in X-ray crystallography, we use X-rays and we pass it through the crystallized protein. So in this, we have to first crystallize the protein, and then we'll pass the X-ray. It will make some diffraction patterns. And depending on the arrangement of electrons in the protein, it will diffract X-ray. And in that way, we obtain the diffraction pattern. And with that diffraction pattern, we will be able to identify, or we will be able to infer the structure of the protein. So in the later sessions, in the later weeks, you will see that how X-ray crystallography help you to visualize the structure of protein. Then comes NMR spectroscopy, which is based on the concept of magnetic resonance. So on the basis of what are the atomic interactions which are present in the protein, we will be able to see the structure of the protein, which is only based on magnetic resonance. And then cryo-electron microscopy. So in this, we use the electron microscope. So firstly, we take the protein and we froze it into the vitreous ice, and then we use the high resolution. We take the high resolution images of that protein with the transmission electron microscope, and with that, we will be able to see the structure of protein. So these are the methods. And in this table, I have defined the strengths and limitations and what kind of proteins are well suited for these particular techniques. So these, if you are interested, you can read this table. And then there are many challenges which are associated with the protein structure determination, such as for X-ray crystallography, you have to perform crystals. So for all proteins, it is difficult to crystallize them. Then these proteins have dynamic nature. That means they have multiple conformations. So with X-ray, you can only analyze its static configuration or static conformation. So dynamic nature is also one of the issue. Then sample requirement, not all proteins can be frozen, or not all proteins have that particular that can support the NMR technique. Then resolution limitations. So there are means for example cryo-EM. So it might not give that in much resolution for certain type of proteins. Then time and cost. So these instruments to perform these type of identifications or structural studies, they are quite costly, and it also take a lot of time. And if there are some post-translational modifications, so that is also difficult. And then computational complexity. So after observing or after getting the X-ray diffraction data, it is also complex to determine the structure. There are multiple things that also undergo. So that interpretation is also challenging. So these are all the challenges which are associated with structural determination. And then but now, all thanks to the AI, artificial intelligence, which came into picture, and it has solved the problem of protein folding. Because previously, if we have a sequence, we if we have a sequence of protein, we can predict that what could be it folded into. But the issue is that means it can take multiple folds. So to decide what fold it will take, it is very difficult. But now the with the help of artificial intelligence, machine learning, all these techniques came into picture, and now they have simplified it. So now they have developed a tool which is known as AlphaFold. So we simply have to give a sequence and it will predict its structure. So now, as you can see that how technology has advanced. So in the cryo-EM or in the X-ray crystallography, people were struggling for years to determine a single protein structure. But with the help of the AI, now we can, in seconds or in minutes, we can predict the structure of a protein. So this is how technology influenced the science or how it is influencing the study. So these people, David Baker, Demis Hassabis, and John M. Jumper, they received Nobel Prize in Chemistry in 2024 for their protein structure prediction as well as computational protein design. Yeah, so this is the server of AlphaFold where you can explore it. You can see that it can predict the structure of a protein, although there are certain limitations also, but yeah, it can perform it with ease for maximum of the proteins. Yeah, so now I have few practice questions which are left and the rest content is cleared. So, uh, are you guys, do you have time so that I can go through these questions? Because it's already 8:04. So if you have time, then we'll go through these questions once. So there are just 10 practice questions and we'll go through them and then I will end the session. So, okay, so, okay, if you will not reply, then I'll go through these questions. Okay, so I want you to answer them. So first question is that transcriptional factors they interact with option A is lipids, option B is DNA, option C is proteins, and option D is sugars. So you have to tell that transcription factors interact with which one of these molecules. So you can type in the chat box and I'll see whether you will be able, whether you were able to follow this. Okay, yeah. So you are right. The transcription factors they interact with DNA. Now the next question is that which one of the following is known as father of genetics? So option A is Charles Darwin, option B is Robert Brown, option C is Mendel, and option D is Walter Fleming. So you can simply type the option also. This we already discussed, and it's the easiest question of this session, I guess. Yeah. Okay, very good. So the right answer is Mendel, and we'll also see the other options as well. So Charles Darwin, so he is known for formulating the theory of evolution by natural selection, and Robert Brown, so he discovered nucleus, and Mendel, he is known as father of genetics, whereas Walter Fleming, he discovered mitosis, and we have also seen that he was a cytogenicist, so he performed the study that how the chromosome moves during the cell division. Yeah, so this was it. And now next question is, the first enzyme to be discovered was option A, diastase, option B, aromatase, option C, carbapenemase, and option D, cellulase. Yeah, so these MCQs are important because they might come in your final exams. So just go through them carefully. Yeah, so right, you are right. So the first enzyme to be discovered was diastase, and it was identified by Anselme Pine. And this enzyme is involved in the breakdown of starch into sugars. So starch are the polysaccharides of sugars. So it break it into smaller sugar subunits. Then if we talk about aromatase, so aromatase converts the androgens to estrogens, but it was not the first one, so it is not the right option. Then about carbapenemase, so it is an enzyme which degrade carbapenem antibiotics. So it degrades these antibiotics, and then cellulase, so it breaks down cellulose. So the right answer was diastase. Now next question is, which of the following is an example of a pentose sugar? So option A sucrose, option B glucose, option C fructose, and option D ribose. Yeah, so you both are right. So the right answer is ribose sugar. It is the pentose sugar. So deoxyribose and ribose are the example of pentose sugar, whereas glucose, fructose, and galactose, they are the hexose sugars. So this hexose means they have 6 carbons. So do not get confused by the pentose or hexose ring. Also make sure that number of carbons will decide whether it is pentose or hexose. Now the next question is, which of the following is an example of a steroid? So option A, phospholipid, option B, stearic acid, option C, cholesterol, and option D, all of these. So you have to tell which one is the example of steroid. Okay, so very good. You all have said it right. It's cholesterol because it comes under the category of steroid because it contains characteristic 4-ring structure, which is typical of a steroid molecule. Along with that, it contains the OH group, due to which it comes under sterols. If we talk about phospholipids, so these are the type of lipids in which there is a phosphate group which is attached to the third, in place of third fatty acid, we have the phosphate group. Then in stearic acid, so it is also a saturated fatty acid. So that's why the correct option was cholesterol. Now, next question is, the glycosidic linkages are found in option A carbohydrates, option B lipids, option C adenine, and option D amino acids. So very good. This is also right. So the glycosidic linkage is observed in carbohydrates. So this is how the glycosidic linkage is formed. So these are two sugar subunits, and this is a glycosidic bond. Whereas in lipids, there are ester bonds. Adenine is a type of a nitrogenous base, and in amino acids, they are linked to one another by peptide bonds. So that's why right option was carbohydrates. Now next question is, beta 1,4 glycosidic bond is found in option A glucose, option B amylose, option C cellulose, and option D fructose. So this we have discussed while we were talking about the polysaccharides. So beta 1,4 glycosidic bond. Any guesses for this? Okay, so let's see. So it is present in the cellulose. So in case of amylose, we have alpha 1,4 glycosidic bond, as well as in amylopectin, we have alpha 1,4, alpha 1,6. So cellulose shows beta 1,4 glycosidic bond. So the right answer was cellulose. Sorry. So the next question is, molecules generated through the action of enzymes are known as option A proteome, option B genome, option C metabolome, and option D transcriptome. Yeah, so the right answer is metabolome, because proteome means the entire set of proteins which are expressed in a cell. Genome means when we are talking about the entire genetic material present in an organism. Transcriptome means when we are talking about the RNA transcripts which are expressed at particular point of time within a particular cell. And metabolome means the metabolites. So metabolites are the products which are produced by the enzymatic reactions. So for examples, the glutamic acid is a metabolite. Lactic acid, which is also produced as a result of metabolic reaction, that is also a metabolite. Methionine, ATP, glucose 6 phosphate, glucose to glucose 6 phosphate, sorry. So this is also a metabolite which is produced. So this comes under metabolome. Now the ninth question is, the chemical breakdown method for gene sequencing is also known as option A Sanger sequencing, option B chain termination method, option C dideoxy method, and option D is none of these. So Sanger sequencing, chain termination, and dideoxy method. These three methods we have These are just a single method. We have already discussed it. Okay. Yeah, so none of these is the right one because chemical breakdown method is also known as Maxam-Gilbert sequencing, and it comes under first generation of DNA sequencing in which there is a chemical cleavage of DNA. So in Sanger sequencing, we use DDNTPs instead of any chemical, but in chemical breakdown, we use chemicals. So these three methods are the same. Now quickly just have a look at Maxam-Gilbert method. So in this, they use different chemicals, and there are four different uh, means reaction mixtures. So one of the chemical, it breaks where the A or G is present, and in the second, if there is G present, and in the third, if C is present. So there are different chemicals such as dimethyl sulfate, piperidine, hydrazine, and then hydrazine with NaCl and hydrazine. So these are different chemicals which break when either A or G is present, when G is present, when simple C is present, or whenever C with C is present. So in that way, different bands will be observed, and based on where the position of the bands, we can get the DNA sequence. But this method is not much used because it involves certain chemicals, and these chemicals can influence the DNA. So in that way, this uh, method was not much favored. Now, our last question is the phase problem is associated with. So option A is X-ray crystallography, option B is NMR spectroscopy, option C is protein purification, and option D is none of these. Okay, so yeah, you both are right. So the right answer is X-ray crystallography, in which we observe the phase problem. Phase problem means when there is difficulty of determining the phase information of diffracted X-rays. So which is essential for reconstructing the three-dimensional structure of the molecule. So the intensity information is obtained from the diffracted patterns, but at what angle they are diffracted, that phase information cannot be captured. So in that way, although there are some techniques to help this phase problem, but this is the limitation in the X-ray crystallography. Whereas in NMR, we directly measure the atomic interactions. So we do not look for the phase angles or other things. And in protein purification also, there is no such phase problem which is observed. Okay, so this was it for today's session. So if you have any doubts, then you can ask, and then I'll end the session. So if you need any help related to assignment or any other information which you think that I should add or something like that. Okay, so if everything is clear, then I'll end the session. So thank you so much for being there. And from next session, I'll make sure that there is not much content in the slides so that we get enough time for discussing the questions instead of talking about the topics. So thank you so much.